Thank you very much. It is a great honor that I can talk about our findings here in this wonderful conference. The title is Quantity over Quality, and I would like to show you that this statement is true not only in many fields of life, but also in anti-tumor immunity. So the role of the immune system is to differentiate between the harmful and the non-harmful substances. They have to tolerate the non-harmful substances. The harmful substances, quite logically, are viruses, helminths, bacteria, fungi, but there are normal and non-harmful such microbes in our bodies too. I can say maybe recently, but around 3 decades ago, it became quite clear that cancer is also a very harmful thing against which our immune system has to defend our body. And we are focusing on the adaptive immune system, the adaptive immune response, which is different from the innate immune system in several things. First, it's highly specific. It means that there is a specific response against a specific molecule. Second, it's very effective. So we can say that in some terms it's even more effective than the innate immune system, and it can effectively destroy these invaders or other harmful substances. On this picture you can see the basic unit of adaptive immune response. This is the so-called immunological synapse. And I would like to introduce you this synapse through the recognition of cancer cells. So on the left side, you can see a cancer cell. And in cancer cells, the DNA is constantly mutated. And as the cancer develops, more and more mutations are accumulated. Several mutations become or doesn't do anything with proteins. But some mutations also modify the amino acid sequence of proteins, so there will be protein change. And if this protein is changed, then these proteins can be degraded, or they are constantly degraded inside the cell to short peptides. These short peptides are usually 9 amino acid long peptides. And these peptides are bound by the so-called HLA molecules. These HLA molecules are essential to make visible these peptides that are generated inside the cell, and they make these peptides visible to the cells of the immune system, here the T cell of the immune system, and this T cell with its receptor, it can recognize the peptide that is presented by the HLA molecule. So if this HLA presentation is not happening, then the tumor cell will be invisible for the T cells. Unfortunately, the cancer cells developed several tricks. For example, they can overexpress molecules with which they can inhibit the activation of T cells. So although there is a modified peptide sequence in the cancer cell, a mutated peptide, and there is a T cell that is specific for this peptide, the T cell will not be activated because the tumor cell overexpresses this PD-1 ligand with which it inhibits the activation of this T cell. And 2 decades ago, it became clear that by blocking this interaction between the PD-L1 and the PD-1, we are able to reactivate the T cells, and it will result in the destruction of tumor cells. And this approach revolutionized oncotherapy. So 2 decades ago, a late-stage melanoma or a late-stage lung cancer was incurable. 95 to 99% of patients died for sure in several years. So on this plot, this is a survival plot, on the horizontal axis, you can see the time which was taken from the initiation of the treatment. And on the vertical axis, you can see the survival probability. The red curve shows you the original treatment, the success of original treatment. You can see that it gets to 0 in around 1.5 years. The new treatment, with this new treatment approach, around half of the patients survive. And we are glad because those patients that survive this year, they are very likely to survive for decades. So maybe we can say that they survived their cancer. Because it was revolutionary, it's quite logic that it was awarded Nobel Prize. James P. Allison and Tasuku Honjo, they made important research in developing this treatment, and they were awarded Nobel Prize a few years ago. However, we have these patients, these around 50% of patients who still do not respond to the treatment. The cancer immunotherapy field tries to find out what can explain the lack of response in these patients. And view governed the field that the higher number of mutations can be found in a cancer sample, it is more likely that the patients will respond to the treatment. Why is that? Because it's much more likely that there will be a modified peptide sequence presented on the surface of the cancer cells. It's much more likely that there will be the T cell inside the sample, which is specific for that peptide, but it cannot be activated. So it's much likely that if we reactivate these T cells, then we will see response to the treatment. However, I would like to show you that it's not really the case. So in our work, we focused on these HLA molecules. So they are the molecules that make visible these modified peptides on the surface of cancer cells. These molecules has extreme diversity. It means it's an extreme genetic diversity. It means that there are many different variants, and as a result, it's unlikely that there is 2 individuals in this room who carry exactly the same HLA variants. So we are quite different in this regard. And different variants have different peptide binding capacities. So it means that a given variant can bind peptide sequences that carry certain amino acids. Other variants can bind other peptides that carry other kind of amino acids. And certain variants are associated with susceptibility or protection from different infections, and others are associated with autoimmunity and allergy. And they have a recently quite neglected role or neglected feature, which was not really explained and examined. This is the promiscuity. The promiscuity of these molecules means that how many different peptide sequences can be bound and presented by a given HLA variant. So there are variants that can bind only a small different number of peptide sequences, and there are variants that can bind magnitudes higher of these peptide sequences. The former ones are called specialists, and the latter ones are called generalists. And why are these generalists present in human populations? In our previous work, we showed that as these generalist HLA molecules are able to bind peptides of more pathogens, they are much more prevalent in those regions in the world where the pathogen diversity is high, for example, in Southeast Asia. So these generalist HLA molecules were selected during evolution in these regions because they can be associated with effective protection against a higher number of different viral and bacterial species. So we asked the question, do carrying generalist molecules result in better anti-tumor immunity? The logic is that these generalist HLA molecules are more likely to bind the mutated peptides that are generated inside the tumor cells than the specialist molecules, so more such mutated peptides will be present on the surface of the cancer cells. So if we reactivate the T cells, it's much more likely that they will respond, these patients will respond to the therapy. So we examine different cancer cohorts, cancer immunotherapy cohorts, and we determined how many generalist alleles and specialist variants are carried by different individuals in these cohorts. And we found exactly the opposite what we expected. So here there are three cohorts, lung cancer cohort, melanoma cohort, and there are two different treatment options, the CTLA inhibition and the PD-1 inhibition. And the blue curve shows the patient group in which the promiscuity of these HLA molecules is low. So they are very likely to carry specialist HLA alleles. And in the red group, there are patients who carry mainly generalist HLA alleles. And as you can see, independent of the treatment and the tumor type, those patients who carry generalist HLA alleles are less likely to survive after the treatment. And we asked why. We had many ideas. Most of them were not correct, and I would like to show you the most likely thing, the most likely explanation. So in order to understand this, we first have to look into what is needed for the effective recognition of a mutated peptide in a tumor cell. So basically, every mutated peptide has its non-mutated counterpart. And these are usually 9 amino acid long peptides, and usually the difference between these peptides, this mutated and normal peptide, is only 1 amino acid. And unfortunately, or of course it's an evolutionary thing, so it's not unfortunate, but T cells are cross-reactive. So it means that T cell is not specific only for a given amino acid sequence, but if it's very similar to that sequence, then the T cell, the same T cell will recognize that sequence. So if the HLA molecule present the original non-mutated counterpart of the mutated peptide, then it's very likely that the same T cell will recognize this and this peptide too. And of course, when there was no cancer, our immune system was trained to tolerate these peptides. And these T cells are usually responds with tolerance to those mutated peptides. That's counterpart, non-mutated counterpart, is also presented by the HLA molecules. And the good distinction is present when only the mutated counterpart of a given peptide is presented to the T cells. And we thought that in the case of generalist molecules, as they are generalists, so they are generally bind many different peptides, it's much more likely that not only the mutated but the original peptide is also presented. While in the case of specialists, we expected the latter case. And we found this. So there were methods and approaches with which we were able to enumerate the distinction capacity of the HLA molecules. So how likely that the original peptide is not presented, but the mutated peptide is presented by the HLA molecule. And on the vertical axis, you can see the HLA promiscuity. So on the right side, there are the generalist molecules, and on the left side, there are the specialist molecules, and on the vertical axis you can see the distinction efficacy. And there was a significant negative correlation between the age-allele promiscuity and the distinction efficacy. And additionally, so if this is the case, we would expect that in those tumor samples where generalist age-allele molecules are present, there are signs of immunological tolerance. So with typing the immune cells, we are able to define whether there is an immunological tolerance or a severe inflammation inside the cancer sample. And it was exactly what we found. So in these tumor samples where generalist molecules were present, so in those individuals who carried generalist molecules, their cancer were associated with immune tolerance in the tumor microenvironment. So as a summary, we can say that, of course, these generalists are in populations because they have a role. So they are evolutionary beneficial because they are associated with better antiviral immunity against a higher number of pathogenic species. But as a negative trade-off, they result in worse anti-tumor immunity. And of course, it's quite logic that cancer was lower selection pressure during evolution. And infection was a much higher selection pressure during evolution. So this thing counterbalanced the anti-tumor immunity negative trade-off. So I would like to thank everybody who helped in this project. And I would like to highlight, I cannot say or cannot express enough my gratitude to the National Academy of Scientist Education. Because I was a junior mentor when it started, and I'm being a mentor now. And those people that are highlighted with red color, they are currently in the program or were in the program. For example, Benjamin, he started in the program, so doing science in the program 8 years ago. And he graduated as a medical doctor, and he came to my group as a PhD student, and now he's doing his PhD. And Anna and Alexandra, they are currently working actively in my research group. So I can say that the program is significantly beneficial for myself too. So thank you, and thank you for the attention.